awesome. Good evening. Thank you so much for having me here tonight. My name is Elena Leander, and I'm a PhD candidate at UT Austin. And I do think a lot about plant and specifically arbuscular mycorrhizal fungi relationships and how they respond to different types of environmental change. And so I was super excited when Angel and April reached out and invited me to give a talk. So I'm just going to share a little bit of background on what mycorrhizal fungi are and then jump into a little bit of my research. So here's what I'm going to cover tonight. We'll start with some basics. What exactly are mycorrhizal fungi? Then I'll introduce you to these specific underground specialist arbuscular mycorrhizal fungi, which I'm going to call AMF for short. And then we're going to walk through why these hidden partners matter so much for ecosystem resilience. And then I'm going to share a little bit of the research that we've been conducting right here in the research lands at the Wildflower Center, looking at how different approaches to restoration can affect these fungal communities. And then lastly, I'll leave you with some practical tips for supporting these type of fungi in your own backyards. Does that sound like a good plan? Yeah. All right, awesome. Let's get started. So first up, what exactly are mycorrhizal fungi? I saw some folks that were nodding their heads like they knew what they were, and some folks not as much. Also, how many folks say fungi and how many say fungi? 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 All right, then I'll do fungi even if it's not exactly correct. Because that's what I say as well, but I think maybe technically it's fungi. All right, so I wanted to look at this word mycorrhizal. And if we break it down, it really comes from two Greek roots, no pun intended, meaning myco, fungus, and then rhiza, root. So it literally means fungus root. So when we talk about mycorrhizae, we're talking about the relationship between the plant root and the fungi. It can't be one without the other. And these mycorrhizal mutualisms are not exactly rare. In fact, the vast majority of land plants, around 90% of plant species on land form mycorrhizal partnerships. So they are absolutely fundamental to how terrestrial ecosystems function. And when you see a plant growing, chances are very high that it is associating with at least some type of mycorrhizal fungi. So there are four main types of mycorrhizal associations, and they differ in how the fungus and the plant interacts. And here you can see some microscopic images of all four types. So we have arbuscular mycorrhizal fungi, upper left. Then we have our ectomycorrhizal fungi, upper right. And then we also have some less common, but also very cool, orchid and ericoid mycorrhizal fungi types. And each of these has evolved to work with different groups of plants, and they all have their own unique structures and functions. Another main way that we kind of categorize the different types of mycorrhizae is whether they colonize inside the root cells or they remain on the outside of the root cell. So the ones on the left, the endomycorrhizae, will go into the root cells, and then the ones on just that one in the upper right, ecto, will remain on the outside and form kind of a sheath around the root and go in between root cells, but never inside. So even though I'm not talking about ectomycorrhizal fungi tonight, after this slide, I wanted to throw this up here because I know we might have some folks in the audience who are really interested in mushrooms. And I do not actually study mushrooms. I don't know that much about mushrooms, but ectomycorrhizal fungi are the ones that form partnerships with many of our trees. So you can think pines or oaks or other woody species. And they're going to be the types of fungi that produce many of the mushrooms that we're familiar with. So chanterelles and porcini and Super Mario Bros. Mushroom. And so this is for you all, for the mushroom folks. And I'm actually now going to talk about Arbuscular mycorrhizal fungi, and even though they don't produce mushrooms, try to convince you that they are also very cool. They do partner with about 80% of land plants, including the Texas native wildflowers you see here, the tick seed, the prairie coneflower, the primrose, the bee balm, and the blanket flower. And they are actually microscopic and invisible to the naked eye, but absolutely essential. So, okay, now that we have a basic understanding of what mycorrhizal fungi are, let's go ahead and get to know our specialists, the arbuscular mycorrhizal fungi, the AMF, in a little bit more detail. So this is like one of my favorite facts about AMF. This relationship is ancient. The relationship between plants and AMF is very ancient, and there's actually fossil evidence that suggests these fungi helped plants move from water to land about 400 million years ago. So just thinking about that for a moment, when plants first started moving from aquatic environments onto land, it's a huge challenge because the terrestrial environment is a lot harsher. There's really intense sunlight, really dry conditions, and nutrients are all locked up in rock and the soil, rather than just being dissolved in water. And so the general consensus hypothesis is that plants could not have made this transition without the fungal partners helping them access nutrients and water in the soil. So in a very real sense, when we look around at the world as we know it, the forests, the grasslands, and all the terrestrial life that depends on plants, we can acknowledge that it exists because of this ancient partnership between plants and fungi. How many people knew about this history? Some folks. It's pretty cool. All right. I also want to break down this term arbuscular. So it comes from the Latin term arbusculum, which means little tree. And when you look at this microscopic image, you can probably see why. These structures that are inside of the plant root cells branch and rebranch, creating these beautiful tree-like structures known as arbuscules. And they are the primary site of resource exchange between plants and fungi. All right, now looking at how that plant AMF relationship works a little more closely. So we have a plant, if we use this primrose as our example, we can see that its roots are going down into the soil, and the AMF will colonize these roots and form a few different types of structures. So first, as we just mentioned, the arbuscules, where nutrients, especially phosphorus and nitrogen, are being exchanged from the fungus to the plant, and they are receiving in return these photosynthates, which are sugars, or essentially carbon dioxide that is being fixed from the atmosphere by the plant through photosynthesis. And then you also have the hyphae, which folks might be familiar with. There are these thread-like structures that extend into the soil, essentially expanding the root system of the plant, sometimes by hundreds times over. And they can go and explore different pore spaces that the roots might simply be too large to access. And then finally, you have these vesicles. So those are those kind of filled in round circles on the image here. And they are storage structures. So the fungus can use these to store nutrients and lipids or fats. And together, the hyphae, vesicles, and arbuscules form this basis of the mycorrhizal symbiosis. Okay, so I wanted to show you a map of the global distribution of AMF, which illustrates predicted mycorrhizal richness across the globe and was created by the Society for the Protection of Underground Networks. Are folks familiar with that group? This is the Underground Atlas is So cool. If you're into the ectomycorrhizal fungi a little bit more, they also have maps that illustrate predicted richness for those type of mycorrhizal fungi. And here we can see that the warmer colors, the yellows and the greens, are indicating areas that are predicted to have higher species richness of mycorrhizal fungi. And so there's some patterns coming up here. You might notice that some of the highest diversity is found in tropical regions, but also in areas dominated by grasslands, including parts of North America, Central Asia, Southern Africa, and Texas. And zooming in on Texas a little closer, on the left, we're seeing that predicted richness. And then on the right, we see a predicted endemism, meaning how many species or taxa of AMF might be unique to Texas. And in Central Texas, we see moderate to high richness and endemism predicted for these AMF, which suggests that there may be fungal taxa here, especially in our unique karst ecosystems and the blackland prairies that are found nowhere else on Earth, which is one of the reasons why understanding and protecting these communities is especially important. All right, so we've met the AMF. And now I want to talk a little bit about why these partners matter so much for ecosystem resilience. So when we think about ecosystem resilience, we can kind of define it by these three key pillars. Resistance, which is the ability of an ecosystem to withstand a disturbance or a change. And then recovery, which is the ability of the ecosystem to kind of bounce back post-disturbance or change. And then this term adaptive capacity, which speaks to the ability of the system to reorganize or adjust to new conditions. And AMF contribute to all three of these pillars in so many ways, but I'm going to talk about just a few. So AMF produce these incredible structures called spores, which I'm thinking maybe folks have heard about before as they relate maybe to other types of fungi, but that's exactly what you're looking at here. So spores are remarkably diverse in terms of size, color, structure, shape, and they can remain dormant in the soil, kind of similar to seeds, creating a spore bank. And even if hyphae are relatively fragile and can be destroyed by different types of disturbances like drought or fire or tillage, the spores are tough and they have these thick walls that can protect them from disturbance. So, audience, which pillar of resilience do we think this trait of AMF belongs to? Okay, resistance. I heard a recovery. It might be a little bit tricky because I feel like it really could be either resistance or recovery. I'm putting it in recovery because after disturbance, the spores can persist. And then once conditions improve, they can germinate, quickly recolonize plants, and help the plant community recover faster. Also resist in that way too, yeah. Okay, so I wanted to talk about the hyphal networks themselves. So the thread-like structures extending through the soil are incredibly efficient at acquiring nutrients, the phosphorus and the nitrogen and the water that can oftentimes be out of reach for the plant, especially here, Central Texas, when we encounter these really dry conditions and we are kind of lying on top of these really rocky, sometimes shallow soils. So which pillar of ecological resilience could this AMF trait belong to? That was Sean. Why would you say that? I'm thinking like the way I organize things is just one way to organize them and interpret them. I actually put it with resistance, but just thinking that this enhanced nutrient and water stress tolerance helps the plants stay intact, the plant communities stay intact and resist these stresses as they come up. So a plant that is well nourished and well hydrated is going to be better able to withstand drought, disease, and other types of challenges, really similar to how we are able to better fight off illness when we're nourished and hydrated. And then lastly, you can see here 3 different root systems with different types of AMF indicated by these different colors. So in a really diverse AMF community, when conditions change, plants can kind of adjust which fungal partners they choose to invest in most. So some AMF taxa might be better at drought tolerance, others might be better at accessing phosphorus, and others might be better in withstanding in disturbed soils. So having this kind of diverse portfolio of potential fungal partners means that the plant community can be flexible in how it responds to changing conditions. And which pillar of ecological resilience might align with this aspect of AMF? That's where I put it, adaptive capacity. So the plant communities are not locked into one strategy, but rather they can shift their partnerships depending on what their current environmental challenge is. And that flexibility is what adaptive capacity is all about. So when we put it all together here, the spore diversity, the extensive hyphal networks, and the flexibility of these different AMF partners and what they have to offer, we kind of get this complete picture of how AMF enhance ecosystem resilience. Okay, now I wanted to share just a little bit about some of the research that we've been conducting here at the Wildflower Center to understand how these fungal communities respond to different restoration approaches. So here's an aerial view of our study site at the Wildflower Center. Have folks been back in the research lands? I'm not burning it. But yes, and they had recent burns. So this experiment, and I'm not even representing all the different types of treatments in this map here, but it has been going on for 25 years, run by staff and volunteers at the Wildflower Center. And they're essentially looking at different types of disturbances. So either mowing or burning in different seasons. Here I'm looking at winter or summer and examining how that affects the plant communities. And grasslands are disturbance adapted systems. So here in Central Texas, they have been maintained historically by lightning strikes and wildfires and indigenous burning practices. And so you cannot have grasslands, and in this case, we are working in juniper oak savannas without disturbances. So here is what a summer burn plot looks like on the ground. You can see it's pretty open. The above ground vegetation is, for the most part, gone, the woodier vegetation. Well, on the right is a control plot that hasn't been burned or mowed. And you can see it's much denser with pretty well-established woody vegetation and lots of leaf litter. And so these are very different environments from a plant's perspective and also from a fungus's perspective. Yeah, absolutely. Well, sorry. No. The one that showed the whole map. Oh, yep. So the control is got higher level vegetation. Is that true? So this is something that we're looking at. And so just from looking at it from this aerial view, it looks like it has denser, woodier vegetation, right? I would agree. And I'll talk a little bit about what kinds of species we found in the different plots. That's a really good observation. So our first overarching research question in this project is, how does restoration approach specifically this combination of disturbance type and application season structure both plant and AMF communities? And we're thinking about this as a system where the restoration approach affects the plant communities, the AMF communities, but we also know, and we've established, that those two things affect each other. So we have to take that interaction into consideration when we are trying to calculate these resilience outcomes. So what we're really asking is, do different management approaches create different plant fungal partnerships? And if so, what does this mean for restoration practitioners who are trying to restore grasslands? All right, I'll walk us through our sampling design. In fall 2024, we collected, so this was last year's fall, we collected 3 major types of data. First, staff and volunteers did their routine plant community surveys. Has anyone participated in those surveys before? It's usually a big team, an awesome team, collecting data across a permanent 50-meter transect in these 1 by 1 meter quadrats. There's a ton of them along that transect. And then we collected bulk soil samples, which are represented by these brown dots right above the transect. And those were collected at each plot and homogenized to represent the heterogeneity across each plot of the soil fungi. And then we collected rhizosphere soils. So bulk soil is representing the community of fungi and the influence of the plant community on those fungi, whereas rhizosphere soils are within just a centimeter at most of a plant's root zone. And so plant identity can have a big, host identity can have a big influence on the kind of fungi that we see in those rhizosphere soils. And I was specifically interested in 3 different grass species. I think a lot about grass. I love wildflowers, but I also think a lot about grass. And first we are looking at, on the left there, the Nacella is a winter grass. It's a dominant native. And then the Bothriocloa iscamum is King Ranch bluestem or yellow bluestem, which is an invasive. And then on the right there, this is an interesting one, the Bothriocloa lageroides is the silver bluestem, and it is a native species, but it's closely related to our invasive grass species. So I'm not representing this here today, but I have some other hypotheses I'd like to test after collecting the rhizosphere soils from those 3 specific grass species. All right, so we're going to start with what we found for our plant communities. So this is what we call a principal coordinate analysis, and it's a way that we can visualize community composition. So in this case, these are our vegetation communities separated by our different treatments. And how you can interpret this is, when you look at a point, it represents a plot, and the plots that are closer together on this graph are more similar in community composition. And the colors show these different restoration treatments, and the shapes show the disturbance type. So we're seeing a really clear separation between treatments with a little bit of overlap. And when we ran the stats on this, we did find a significant interaction of disturbance type and application season, meaning that burn versus mow has different effects depending on plant community structure, depending on what season you apply them in. And you can kind of see we have these summer burns that are clustering a little bit closer to the bottom near our winter burns. And then we have our a little bit of a more overlap between the control and the summer mow, with these winter mows somewhere in the middle. And so this is great. It's really cool to test this statistically, to know that the treatments are doing something in terms of restructuring plant communities. But if we want to know what's really driving those patterns, we can do an indicator species analysis and ask which plants are statistically significantly associated with specific treatments. So this goes back to what someone was mentioning earlier, where we have different types of species that are associated with the different types of treatments. And one big pattern that we found is that, yes, a lot of the woody species are found in control, winter mow, and summer mow plots, which might make sense because we're not getting that same kind of top kill that we would expect from a fire. But what we also found is that in summer burns specifically, we have a number of species that only are significantly associated with that treatment, which means that we might, with summer burns, create opportunities for more fire-adapted herbaceous species, especially because in the summer, those burns tend to be a bit more intense due to the higher temperatures. So this might be useful information for land managers if your goal is to maintain an open grassland system and prevent woody encroachment. Burning, especially summer burning, appears to be more effective than mowing. All right, now looking at, I will say, with the vegetation data, the Wildflower Center staff and volunteers has been collecting this for like 25 years. There's a whole bunch of data. They've published work from it, and they're also still analyzing some of this. So this was just this one snapshot of time, fall 2024. Looking at the fungi, also a snapshot, but something that we can match up with that fall 2024 vegetation data. I first looked at diversity using the Shannon Diversity Index, and I separated the different soil types into bulk versus rhizosphere and by plant host identity. And on the left, we see a clear pattern where disturbed plots, whether those be burn or mow, have higher AMF diversity than our control plots. And this might be a bit surprising at first, but it makes sense. For one, we have more herbaceous species in our disturbance plots, and we have more AMF associating with herbaceous species versus woody species. And also, when we have disturbances, it creates a little bit of heterogeneity in the environment, which can support more diverse fungal communities. It can also prevent any one fungal taxa from becoming too dominant. And then on the right here, we have our rhizosphere soil samples from the 3 host plant types. And I think what's fascinating or what's interesting is that diversity differs significantly by host plant, but not by restoration treatment. And we see that the invasive species has somewhat lower fungal diversity in its rhizosphere, which is in line with previous published research relative to native plants. But within each plant type, we're not seeing that restoration approach actually has a strong effect. So what this suggests is that in bulk soil, disturbance increases diversity, but in the rhizosphere, that soil zone that's so close to the plant's roots, it's plant identity that matters the most. Plants are selecting their fungal partners, and that selection is stronger than the effects of how the land might be managed. Sorry. All right. So we've seen these type of graphs, these types of figures before, looking again at community composition, but this time for the AMF. In the bulk soil on the left, you see this really clear separation by disturbance type and season, similar to what we saw in our vegetation communities. And in the rhizosphere on the right, the pattern is a little bit more complex. So we are still seeing the significant effects of disturbance type and application season independently, but now host plant also matters. And so in the rhizosphere, we really have that interplay represented in our previous graph between what the plant is doing, what the plant is selecting, and then the environmental filter created by management practices. Both matter, neither is overriding the other. So summarizing those things, and we're still, I'm still analyzing the data, and I actually also have the ectomycorrhizal fungi data as well. So we're hoping to find all sorts of cool patterns from moving this project forward. But first, we found that restoration approach does matter for both plant and fungal communities, and the combination of disturbance type and application season. So burn versus mow, summer versus winter, creates these really different ecological conditions that can select for different types of communities. And second, host plant identity is a really strong driver of those rhizosphere fungal communities. Even within the same management and treatment, different plants can associate with different fungi. And that might make sense given what we know about plant fungal specificity. Certain fungi really like associating with certain plants, but it's also cool to see it documented here in this real world restoration context. And third, and most broadly, our findings suggest that restoration management can support healthy plant and fungal relationships. The disturbances that are being applied aren't destroying these systems, they are reshaping them in ways that may actually enhance diversity and function, which is encouraging for restoration practitioners. All right, now I'd like to leave you, not that I am the expert, but just with some practical advice for supporting these types of fungal partners in your own yards, gardens, backyards, in your own gardening endeavors. So AMF dues. First of all, I think plant native seed mixes, which are being handed out tonight. Were handed out tonight. Yes. Can really encourage AMF partnerships, especially if you know which ones are highly mycorrhizal or are dependent on AMF. And secondly, go easy on the fertilizer and the fungicide. That might be really obvious with fungicide because it literally means fungi killer. But with the fertilizer, when you dump on a whole bunch of fertilizer, especially if it's high in phosphorus, the plant thinks, oh, well, it doesn't think, maybe it feels whatever. The plant does not invest as much into these mycorrhizal fungi relationships because it doesn't need to, because it's already getting that input from the fertilizer. And third, disturb smart, which is what some of our preliminary research findings point to. Some disturbance might be good for AMF diversity. It might prevent, dominance by a few species or taxa and create heterogeneity in the environment, but varying your disturbances. So in a yard context, maybe leaving little patches where you are not mowing, or if you are burning your yard or whatever it is, burning in patches, or if you are a land manager on a larger land area, essentially trying to create this mosaic of different conditions will help encourage diverse plant species and diverse mycorrhizal fungi. And fourth, protect a soil crumb, which I didn't speak to this, but these arbuscular mycorrhizal fungi in particular produce a soil protein known as glomalin, and it's really important for giving the soil its structure, as well as the hyphae are also really important in doing that. And that can prevent erosion, it helps with nutrient cycling, it helps soil carbon sequestration, all sorts of cool ecosystem function stuff. So watching out for heavy tilling or a lot of compaction, because both of those things can be detrimental to arbuscular mycorrhizal fungi. And with that, be patient. Building networks takes time. And that's the end of my talk. I wanted to say thank you, first of all, to the Central Texas Mycological Society for inviting me here, especially Angel and April. Thank you so much. This is such a cool chance to get to share with you all. And then my PI, Dr. Amy Wolf, and also Dr. Sean Griffin, who works here at the Wildflower Center. Of course, the Wildflower Center, all its staff and volunteers who have maintained the treatments and also just been so awesome in so many ways. And then the Society for the Protection of Underground Networks, who funded a lot of this work for me. And yeah, I'm happy to take any questions I think we have a little time to chat. So that's all. Thank you. And thank you all so much for being here tonight on this evening, last Tuesday twilight of the season. So now it's to you for questions. Yeah.
The FDA used to send me surveys as if I was a no-till farmer.
Okay.
One of the things that they're reading is don't pull up your plant roots, cut them off, and leave them in there.
Yeah.
To me, that's a neat idea, kind of, except for the fact that now I have a structure in the way of a new plant. I have to plant between the other two plants, and I have tons of space. But the no-till part, I think, is pushing this to keep the fungus in place, right?
Yeah.
So how do we, as small time gardeners, we grow our plants or do we come off?
Yeah, that's a good, I mean, you bring up a good point. So I guess just repeating the question here is, and you correct me if I'm misrepresenting this, but what kind of are the benefits in gardening to leaving plant roots in place versus pulling out the whole plant once it dies? And so when you leave the plant roots in place, they can serve as reservoirs for these arbuscular mycorrhizal fungi, especially since a lot of them are, well, all of them are obligate biotrophs, meaning they cannot grow, reproduce on their own without living inside of a plant. And so also the plant roots, similar to the hyphae of the mycorrhizal fungi, can provide a lot of structure for the soil as well, serve as mycorrhizal fungi reservoirs, and pulling them out, I could see that being useful in certain cases where you are ready to plant something new, or you are worried about pathogen buildup. So maybe it's a little bit context dependent, but those are the benefits of leaving roots in place. And if you can do it, I would say, yeah, go for it.
We have a question from the online audience. So Elizabeth Gray asks, what do you recommend for a newly developed parcel of property where all the existing plants have been scraped and the dirt is very compacted, native plants, lots and lots of compost mulch, and question mark?
Native plants, lots of compost. So all of the plants have been removed and so it's really bare, and then this person is adding native plants, compost, mulch.
That's like kind of the question. Do I just add a lot of compost and native plants?
Yeah, I mean, again, like I am not, the expert on this, but I would say that it already sounds like, having the mulch there is going to help retain water, having the compost add a little bit of nutrients back to the soil. If you removed all that biomass, or if it was removed and it's really bare, it could be the case that the plants were taking up a lot of nutrients, and then you take that biomass out so it couldn't go back in the soil. I think that, like, what we found is that the rhizosphere the effects of plant host identity on the rhizosphere is really strong. So if you can be mindful in your native plant seeding mix of which species you're planting and whether or not they associate strongly with mycorrhizal fungi, then you might be able to get a little jump start on promoting some of those mycorrhizal fungi in your system so they can build back some of that soil structure and help kind of jumpstart some of that nutrient cycling. So that would be something I would add, is just being mindful about who you have in your seeding mix that are mycorrhizal partners. Yeah.
The same way that we've got these native seed mixes, are there like native spore mixes we might also just incorporate into the content?
Yeah, that's a really cool area of research. I think a lot about Liz Koziol, who works in grasslands and kind of puts together these different relatively native, regional, biodiverse inoculums. With mycorrhizal fungi inoculums, in some cases, you can get commercial mixes which aren't necessarily super biodiverse. They're not necessarily going to be native to your system. And if they are not native to your system, then you are losing out on kind of harnessing that power of those co-evolved relationships between the native plant species and the locally native arbuscular mycorrhizal fungi. And sometimes inoculums, if they're produced in a lab, they can cultivate whatever fungi grow really fast in the lab context, which might not necessarily be what's best for your system. So I would say yes, but with a little bit of caution. Yeah, definitely.
Methods that we use for the research that we're doing, where it's a trap culture method, where you collect a little bit of soil, then you grow some grasses, and then you use that and then you kill off the grasses, and what happens is it sporulates underneath. All the carbon from the grasses goes down into the mycorrhiza and into the source, and so it's like, okay, now I need to actually find a way to exist, and so it just goes into the mycorrhiza. So we have some literature outside that you can grab that might be helpful for that. Yeah.
Awesome. Yeah, and those trap culture methods. Yeah, it's cool. Yeah.
Question is, would you say that the networks might drive the plants, or the plants drive the network?
Oh, it's a great question. And yes, so the question is, are the plants driving the fungal response or the fungal network response, or is it the other way around? And this is debated in the literature. It's like a driver versus passenger hypothesis. And I personally think it's definitely a little bit of both, and it's context dependent, which I know isn't a great answer, but it is folks tend to kind of choose one or the other, depending on if they're more fungi people or more plant people, maybe. But I think it's a little bit of both. Yeah. Yeah.
First off, this is an amazing presentation. I should say it's really interesting. It is not accessible to a normal person. Like an average everyday rancher landowner is not going to be interested in grass. Are you going to work with ag extension? Because those people who are land managers are going to be really interested in the benefits that this would provide to the property. What is your plain language option for this?
Plain language about talking about mycorrhizal fungi in general. I would love, that would be so cool. I would love that. I mean, I do definitely think about how this applies in an ag context, and specifically with arbuscular mycorrhizal fungi, because they associate with so many crop species. I like thinking about that. And so I think developing that kind of language that allows me to connect with and learn from folks in the ag world is something that I'm gonna continue working on. I don't think it's totally, I don't think it's a stretch at all, actually. I think, I've heard different types of language around different inoculums being like, biotic inputs. Actually, some of my family is in the ag world, and my uncle said, biotic inputs are all the rage these days. So I'm kind of also learning a little bit from those folks about how to have those conversations. But I would love to work with an ag extension office. Okay, awesome. Sounds good.
Species, Texas, like the
Yeah.
native species for the actual scales.
Yeah, there are different resources online. You mean that, so the question is there a chart for Texas native plants that shows how strongly they relate with mycorrhizal fungi? Is that right?
Right.
Yeah.
Like your top 10 to get it started and then.
Actually, you know what? I've been looking at a few different sources because I'm trying to incorporate in these analyses different plant traits and also mycorrhizal AMF traits. And I was looking online at a few different sources to get an idea of how strongly mycorrhizal they are, or a measure of what we call mycorrhizal responsiveness, how often they create these associations. And actually, I think the data is a little sparse for Texas, but one that comes to mind is like the AMF trait database, AM trait database. But it would be considered a trait, mycorrhizal responsiveness. And I'm not sure how strong that database, the coverage on that database is for Texas, but yes, it is out there. Or if it's not, it should be, and people will continue to work on it. Yeah.
Like a good segue into another online question. The person asked, like, in this study, were the fungi that were found native to the area, to this area, or is there a way that AMF is getting classified as like native or bioregional? Like
Yeah.
how is that determined?
It's such a good question. And I mean, especially because the AMF are so small, and we are just now really having the tools, the sequencing tools, and that are becoming more accessible to start identifying different taxa. And I say taxa because a lot of times we don't even know we can't even define species for AMF. So I didn't include it here, but I kind of have this breakdown of okay, what genera are these AMF that we're finding out in the soils out here? And we can know enough to know that they're different species or different taxa, but they kind of get lumped under these genus level assignments because our reference databases or what we know about AMF species or taxa in general is kind of sparse. Again, because they're small, but that information is growing. So long-winded way of saying no, that's actually like a really active area of research, kind of this designation of native versus not, and you have to look at phylogenies. But I think in general, doing things like, trap culture methods or taking inoculum sources from really healthy grassland communities or plant communities, using those as your references for what is native, what is local, what is helpful to the locally native flora are the cues that we take in terms of what's native, what's not, what's good for the plants, what's not. Yeah.
Restoration system of fire. I'm specifically interested in like the plant space or even grassland space. Whether that's like controlled burns or what could that look like?
Yeah. Oh, people do all sorts of cool stuff, and it's like really, I mean, yeah, prescribed burns, that's a tricky one if you're in a park space. Mowing, I mean, just exactly this, thinking about a disturbance and really thinking about seasonal timing of that disturbance. So mowing during the summer, I've seen that's really helpful if you have these warm season grasses that are invasive because it knocks them down before the seed heads come up. I've seen folks, do an inoculated plug plantings or do different things like, you know, spreading out trash bags to solarize certain species if they don't want them in the area. And so there's like, just, I've learned from some awesome restoration practitioners, but it really like, it kind of seems like the different opportunities or the different ways you could go about restoring your system are endless a little bit, or like there's a lot of room for exploration and creativity there. But yeah, those are the main ones. I think mowing, burning, solarizing, inoculating, plug, seeding, those kinds of things. Yeah. Did you have a question?
Yeah. That's starting the slow slog of, they have acquired a dump site. They're going to clear the dump site and they want to remediate it. It seems like maybe investigating native plant relationships with these AMF could be beneficial in remediating that soil.
Yeah.
Obviously, I don't want to like plant anything edible there, but like they want to introduce natives and like help turn this huge, it's a big plot, it's like acres, but like, you know, bring back the native species and help kind of like make that
Yeah.
nature sanctuary. And so I'm just wondering, like, has
Yeah.
anyone done any research on these relationships in relation to the remediating and dump site, which kind of was like kind of its own case, but.
I don't know that much about AMF and remediation, but I know that there are some really cool fungi that more so saprotrophic, so these decomposing fungi that can be huge in bioremediation efforts.
You're like,
Yeah.
the mycologist that we worked with to do the trap culture workshops, Danielle Stevenson, she's done several remediation projects using native plants and like spent mushroom substrate, like these saprophytic fungi to help kind of pull up the heavy metals. So what she essentially did was just go and find out like what plants are already growing there that are bioaccumulators of heavy metals and toxins, and then populate those. And they're like the plants that are already usually growing in margins in these like highly polluted areas already. So it's kind of like just following what nature is already doing. And then adding your organic matter, which is like the spent mushroom substrate, also especially pleurotus, oyster mushrooms, like it's a really good with, and there's a lot of debate in the science community if pleurotus is actually helping with the bioaccumulation of heavy metals, but it's still up in the air. There's a lot of like studies you can kind of look at where everyone's saying yay, nay, you know, there's nothing like super clear about that species, but yeah.
Yes.
What is the overall application of fertilizer?
Yes.
And we heard that this AMF putting too much compost out.
So can we hurt AMF by putting too much compost out? I think that it depends on what system you're working in. If you're working with native plants that are used to being in not super composted or nutrient-rich soils, and you're trying to encourage the plant AMF relationships, I actually think, yeah, I think it can discourage the formation of some of those relationships. I've heard in general, you know, if you're planting native plants, and maybe you've heard this too, kind of mix some of the native soil that's less nutrient rich with a little bit of the compost. I don't know exact specifics on how compost and what level of compost might influence plant AMF relationships, but I could expect that, I expect that it would have some discouraging effect of plant AMF relationships if you're working with plants that aren't used to high nutrient, really nutrient-rich soils. Yes.
What's your favorite grass?
My favorite grass? I was just thinking of one not even in Texas, sorry, but let me, I honestly really love the silver blue stem because it's so beautiful when it kind of dries and it has those little silver tufts. And my good friend taught me it smells like grape sweet tarts if you know, smell those seeds. So that's a plus. So thank you for asking. I think, should we?
Should we do one last question?
Okay, one last question. Yes.
actually kind of build on the twofold questions. So the first one, what motivated you to kind of pick grass and wildflowers as like the group of plants that you want to research this introduction, right? Because you mentioned there's interaction happening as well with crops. So like you could have chosen that, investigate that relationship they went with wildflowers. So just wanted to know why.
So why did I choose wildflowers and grasses instead of crops? I think in part because I'm really interested in this restoration piece, this ecosystem restoration piece. And a lot of that has to do with, native grasses, native wildflowers and forbs versus crops. But I'm also really interested in the food production piece. So I think in part it was where I ended up for school, and my advisor and my lab group and their interests as well. And I just I'm not closing any doors one way or the other. But in this case, in my PhD work, I just ended up really kind of incorporating this restoration aspect. And so I think it led me a little bit down that path.
From like your listening experience, do you find that these findings here in this project would also apply to crops, or are they like completely different groups that would require
It's-
completely different testing?
Do these findings apply to crops, or are they two different of groups? It is such an interesting question. Like, I am so curious about that because especially with crops that we, breed to be certain ways, to maybe actually be more dependent on fertilizers or produce really big fruits or whatever it is, sometimes it can discourage these mycorrhizal relationships. And so what I'm interested in the ag context is how can we kind of bring not just inoculum, but this consideration of mycorrhizal fungi, our muscular mycorrhizal fungi, back into our understanding of what traits are important to breed for, especially what plant traits, especially when it comes to building really resilient ag systems. Yeah. Awesome. Yeah, great questions. Thank you all so much. Thank you so much.
Thanks so much, and come by our table and grab our one-sheeter we have on growing your own mycorrhizal fungi using trap culture method. And we also have blocks. So please take mushroom blocks to use to kill your lawn, do all the things that we should be doing, sheet mulching. But yeah, I appreciate everyone coming out. Thank you so much to the Wildflower Center for having us.
It's great. Thank you, Delaney. I had no idea it was not a huge